outline for my talk today, I'm going to give a brief introduction to coral reefs and talk about top-down and bottom-up processes that I thought about, which were specifically thinking about the role of herbivorous fishes and nutrients. And then I'm going to tell you about 3 projects that I was involved in during my PhD. 2 of them are related to herbivorous fish communities and reef resilience, and then I'll tell you about a 3rd project thinking about nutrient impacts on algal communities. And then at the end, I'll spend just a couple minutes talking about the projects that I'm involved in here as a postdoc. So coral reefs are very ecologically and economically important. They're important for fisheries, tourism, shoreline protection, education, and culture. And they are also important because they serve as biodiversity hotspots. And on coral reefs, coral and algae are 2 of the dominant benthic space holders on reefs. And as such, they compete for similar resources such as benthic space, light, and nutrients. And on coral reefs, you can find a variety of types of different algae, and the different types of algae that are present will interact with coral and function differently in a reef system. And so I'm just going to introduce you to 2 broad groupings of algae, which are turf algae and macroalgae. And so turf algae are short filamentous algae that are less than 2 centimeters in height, whereas macroalgae are larger algae that are greater than 2 centimeters in height. And the macroalgae that you can find on coral reefs are very diverse. So here's just some photos showing you a variety of the different macroalgae that you can find on coral reefs. And these are common macroalgae that I found on the reefs that I worked on during my PhD. So in terms of thinking about bottom-up processes on reefs, again, coral and algae compete for similar resources, and these resources then control the growth of coral and algae. Thus, they are exhibiting bottom-up control in the system. But humans are modifying the availability of these resources in coral reef environments, and I specifically thought a lot about human modification of the availability of nutrients in these reef systems. And we're seeing increased nutrients in reefs, often due to runoff from land. And with these changes in availability of nutrients, this could then alter the competitive dynamics that you may see on reefs between coral and algae, but also between the different types of algae that you find on reefs. On the other side, so thinking about the top-down processes, here I was really focused on herbivorous fishes on coral reefs. And so again, we have coral and algae that compete for resources. And then we have the herbivorous fishes present on reefs that consume the algae. And thus, herbivores are exhibiting top-down control on algae on reefs. And the process of herbivory is very critical on coral reefs because it actually helps give coral a competitive edge over algae, which is important because algae is typically faster growing than coral. And so as I mentioned, the algae that you can find are very diverse. So you need the corresponding diversity of herbivorous fishes present on reefs as well in order to exert that control on the variety of algae that you can find on reefs. And so when thinking about a diversity of herbivores present on reefs, it can be helpful to think about herbivore functional groups. So I'm just going to introduce you to those. So these are 4 herbivore functional groups based upon Green and Bellwood from 2009. And so the top 3 groups here, which are the scrapers / small excavators, the large excavators / bioeroders, and the grazers / detritivores, all feed on turf algae. And then the differences between those 3 groups relate to whether or not when they take bites, whether or not they affect the substrate beneath the turf algae that they're feeding on, whether or not they contribute to bioerosion. And then for the excavators, there's a division based upon their size. And then the 4th group of herbivores are browsers. And those are those fishes that are known to feed on macroalgae. And so as with bottom-up processes and nutrients, we see that humans are also modifying top-down processes in reefs by modifying herbivore communities that you can find through the active fishing. And by changing the herbivore communities present on reefs, this can thus change the top-down control of algae on coral reefs. So coral reefs are experiencing a variety of anthropogenic impacts, including climate change, nutrient inputs, and fishing pressure. And these are modifying the top-down and bottom-up processes within reef systems. So we are seeing a lot of reefs around the world undergoing these shifts from coral-dominated states to algal-dominated states. And this has spurred a lot of research into understanding why these shifts are occurring and what can contribute to coral reef resilience to resisting these shifts. And so this is what my research focused on during my PhD. We know and understand that anthropogenic impacts are changing top-down and bottom-up processes. And I really then was kind of asking, well, what are the effects and implications of these changes for coral reef resilience? And I use the definition of resilience, which is resistance to and recovery from disturbance. Okay, so I did my research on coral reefs around the island of Moorea, which is located in French Polynesia. So if you look at this map up here, this yellow square up here is where we are located, and this yellow square way over here is where Moorea is located. And then I have an image of it from Google Earth shown down here. And so Moorea is a long-term ecological research site. So there's a variety of long-term data sets on various biological and physical variables that you can look at publicly. And the coral reefs around Moorea have experienced a number of different disturbances over the years. Some of the recent notable ones include a acanthaster plank eye outbreak in 2007, Cyclone Ali in 2010, and there have been a few coral bleaching episodes in recent years. And for my research, I specifically worked on fringing reefs around Moorea. So those are those coral reefs that are closest to shore. And I worked on a couple different fringing reefs on the north shore of Moorea, which are indicated by this box here. Okay, so now I'm going to tell you about my 3 different projects, and this is the slide that I'll return to in between each of the projects that I'm going to talk to you about today. And I'm just also introducing these cartoon images of herbivorous fish and different types of algae, so representing macroalgae and turf algae here, because I will periodically use these cartoon images throughout my presentation. And so the first project that I'm going to talk to you about is one of the first ones that I did during my PhD, where I was really interested in just assessing the functional roles of herbivorous fishes. And so after my presentation today, if you want to go read the full paper, you can go check it out there. And then also just wanted to acknowledge my collaborators shown along the bottom there. And so, as I mentioned, the functional roles of herbivorous fishes are important for coral reef health because they exhibit that strong top-down control on algae on coral reefs. And when you're thinking about an herbivorous fish community on reefs, you want to think about the functional redundancy and functional complementarity in the system. So I'm just going to explain those concepts. So here with the fishes, the color of the fishes is meant to represent the algae that they are feeding on. So these orange fishes are all, this is indicating that they feed on this turf algae indicated in orange down here. This is just a rainbow fish indicating that it feeds on a variety of algae. And so functional redundancy is when you have a variety of different species that are performing the same function. So when you look at this fish community, you can see that it's fairly redundant in terms of feeding on turf algae because you have multiple fishes that are orange. On the other side, functional complementarity is when you have species that perform different functions that are complementary to each other. So in this case, when you look at this fish community, you can see that the different fishes are performing different functions in terms of they are consuming different types of algae found in this reef system. So this is a community that has a lot of functional complementarity. And ideally, you want herbivorous fish communities that are both functionally redundant and complementary because you want all functional roles to be filled in the system. But the level of redundancy is also important because if you have a disturbance come through and it removes some fish species from the system, if you have other species that were redundant, that function will still be performed even though you've lost some species from the system. And so understanding the level of functional redundancy and complementarity in herbivorous fish communities can be important for assessing and understanding the health and resilience of coral reefs. So that was my objective, was just to assess the functional diversity of herbivorous fishes feeding on macroalgae on a fringing coral reef in Moorea. And so one note is that I specifically focused on macroalgae for the study. And this was because other studies had been coming out recently that were suggesting that grouping all herbivores that feed on macroalgae in one category of browsers may be too coarse, and it may be important to actually subdivide that group into other smaller functional groupings. And also, some studies were also finding that there were specific browser species that were especially important in removing macroalgae on coral reefs. And so identifying those specific species may be important if you're thinking about trying to protect and manage those fish species so they can continue to play the role of removing macroalgae on reefs. And there were a number of other studies coming out at the time that were doing the same thing on reefs around the world. But at the time that I did this, no one had specifically assessed the herbivore communities on reefs around Moorea. And so I did this by deploying macroalgal choice assays with 7 different species of macroalgae pictured along the bottom here. And when I deployed my choice assays, I deployed them with an underwater video camera. So then I could see which fishes were coming and taking bites on which of the macroalgae that were in the choice assay. And so from these videos, I collected standard data such as identifying the fish, getting an estimate of their size, and then counting the number of bites that they took on the different macroalgal species. And then I also calculated Strauss's selectivity index so that I could get an idea of the preference or avoidance of different macroalgal species by the different fish species that I observed. And so here's just one figure of one of the fish species that we observed. I'm just going to go through the setup of this figure before I show you all of them. So this was a parrotfish that we observed. And so I have my 7 macroalgal species along the x-axis, and then I'm showing you the selectivity index values on the y-axis. And so if a value is greater than 0, and if the error bar does not overlap with 0, that indicates preference. And if a value is below 0 and the error bar doesn't overlap with 0, that indicates avoidance. And so in this case, for this parrotfish, we see that it exhibited a preference for 2 of the 7 macroalgal species shown here with the pink boxes. And so overall, we observed 7 different fish species coming and taking bites on our macroalgae. And so the figures are set up the same way as what I just showed you, but I've also color coded the fish species that we observed based upon the functional groups that I introduced earlier. Now, one note about the functional groups is that we didn't actually observe any of the large excavators / biorotors. None of our species that came fell into that category. So this purple fish over here is actually a triggerfish, which is not classified because they aren't typically thought of as being herbivorous, but we had triggerfishes coming and taking many bites on our macroalgae, so we included them in our data set. And so when you're looking at this, we're thinking about functional redundancy and complementarity. So we were wondering, which fish species could be considered similar to each other. And if you were going to divide this community into different groups, how many different groups might you divide it into? And so we did this by doing a cluster analysis. And so here are the results from the cluster analysis. So again, the names of the fish species are color coded according to their functional groups. And then you can see from the cluster analysis that it indicated we had 2 groups of fish species. So indicated by these black lines up here versus these gray lines down here. And so I'm just going to go back to showing you these figures, but now I've added boxes around those fish species that group together. And so when you're looking at this and trying to understand why the different fish species group together based upon this cluster analysis, it's helpful to get an idea of what their functional groups are, but it can also be helpful to look at what macroalgae selectivities were being indicated for different fish species. So I've just added boxes around all the instances where fishes preferred a certain macroalga. And so what we can see here is that those fishes that are previously classified as browsers, they all group together on this reef. And we actually did not find support for subdividing them into finer scale divisions. And then in terms of the other grouping of our 4 fish species, that kind of was like an other group. So those fishes that weren't classified as browsers. And one thing that may indicate why they also group together is that they seem to just be generally less selective of macroalgae. So only 2 of the 4 species exhibited a single preference at all. And if you look at the over bars and how many of the error bars overlap with 0, you see kind of what I thought of as like a lot of ambivalence in terms of which macroalgae these fishes were taking bites on. So another way you can look at this data is thinking about, well, which macroalgae are being consumed in the system? And so again, if we're looking at those macroalgae where there are preferences being exhibited for them, we can see that there is high redundancy for this first macroalga, which is Podyna. So we have 3 fish species that exhibited a preference for Podyna. And then also for Sargassum, which is this 4th bar from the left, we can see 1, 2, and 3 fish species also exhibited strong preference for Sargassum. So this suggests that there's fairly high redundancy in terms of consuming these macroalgae in this system. And then across the other macroalgae, there seems to be low complementarity because we don't really see any preferences for the other 5 different macroalgae, except for this one. The triggerfish really liked this macroalga, Acanthophorus spicifera. So in summary, our data, based upon the cluster analysis, suggested that we had 2 clusters of herbivorous fishes on this reef. The first cluster were those that were classified as browsers, and they were highly selective of Podyna and Sargassum. And then our second cluster of fishes appeared to be just kind of less selective overall on macroalgae. And in terms of which macroalgae are being consumed and what functions are being performed on this reef, again, we saw preferences were exhibited only for 3 of the 7 macroalgae. And the 4 remaining macroalgae were not preferentially consumed by any of the herbivores. And so when we're then thinking what this means in terms of functional redundancy and complementarity, we concluded that we found more redundancy than complementarity in this reef system. And this suggests that this reef system may be resistant to disturbances that remove some fish species. But because of the low complementarity, it suggests that maybe all of the functional roles are not being filled. And then when thinking about the roles of specific herbivorous fishes, we actually found that fish species that we observed on our reef did not perform the same function in our system as we may have expected based upon previous studies. So as I mentioned, studies like this had been being done in other reefs around the world. And so in some cases, we saw that like fish A in our reef was a redfish, so it consumed this type of macroalgae. But when that fish was observed in a different reef somewhere else, it consumed different macroalgae. So this just suggested to us that we may need to actually assess the functional roles of herbivorous fish species within specific reef systems and not make assumptions based upon their roles, based upon studies done in other places. Okay, so that was the first study I wanted to tell you about. And now I'm going to tell you about another study, which is also thinking about the role of herbivorous fishes in reef systems. But in this case, I'm also going to think about how fishing may impact herbivorous fish communities. And so really thinking that if fishing is altering the herbivore communities that are present on coral reefs, how is that then affecting the resilience of those reef systems? And so again, we're seeing that reefs are shifting from these coral-dominated to these algal-dominated states, which has spurred a lot of research into asking what influences reef resilience and also what kinds of shifts are coral reefs experiencing. And so knowing the type of shift that an ecosystem experiences is critical in order to effectively manage those systems. So in some cases, as an environmental condition, so if you think about like fishing pressure, nutrient pollution, as that condition gradually changes or worsens, you may see that an ecosystem state also gradually changes. On the other hand, you may see a threshold transition where as an environmental condition gradually changes, the system stays in a relatively stable state until it hits a threshold, and then it rapidly changes to a different state. And then finally, you may see in some systems evidence of bifurcation curves, which are indicative of alternative stable states. And alternative stable states are especially challenging for management of ecosystems due to their underlying properties of bistability and hysteresis. So I'm going to explain those concepts real quick. So bistability is when for a single level of an environmental condition, the system can exist in one of 2 possible stable states. And you can imagine how that could be challenging for management efforts, because if you are managing a system and you are managing it to get down to this environmental condition, say this level of nutrients, that's going to be the maximum we have in the system. However, if your system is exhibiting alternative stable states, you don't necessarily know whether your system is going to be in this state or in this state. And as humans, we typically have one state that is more desired than the other. So I will also talk about these in terms of like a healthy versus a degraded state. And so hysteresis then is really thinking about where the tipping points occur when you're transitioning from a healthy to a degraded state or from a degraded back to a healthy state. So I'll just step through that a little bit more. So in this case, if we look at if you are starting with a healthy ecosystem and environmental conditions are steadily worsening, you can see where the tipping point is, where that system shifts from a healthy state to a degraded state. But on the other hand, if you then start with like a degraded system and you look at where the tipping point is, whereas you're improving environmental conditions and where it's then able to switch from the degraded state back up to the healthy state, the tipping point may be over here. And so when you look at those two tipping points, if they don't overlap, that means that you have hysteresis in your system. And so again, you can imagine that this is difficult for management because you may have had a nice healthy state up until here, and then you had a degraded state, and you want to recover that healthy state. But instead of only being able to reduce the environmental conditions to this level, you actually have to improve the environmental conditions all the way back over here before the system will then be able to shift from the degraded state and recover back to that healthy state. And so the functional diversity of herbivorous fishes and the importance of it is well documented in reef systems. But I was finding that previous models that were examining coral reef resilience didn't seem to be incorporating herbivore functional groups into them. So that was my objective for this study, was to incorporate herbivore functional groups into a coral reef resilience model and see if it had any impact in terms of what we see for reef resilience. And so the specific research question I'm going to tell you about from this project was asking, how does variation in the dominant herbivore functional group influence the occurrence of alternative stable states in response to fishing pressure? And so for this study, I focused on grazers, which are those fishes that consume turf algae, and browsers, which are those that consume macroalgae. And then I also included a group of generalists, which are fishes that feed on both turf and macroalgae, because complete specialization is not always the case in coral reef systems. And so to address this question, I did it with a model, and I started with this basic model structure, which I adapted from Van de Lippe et al. From 2016. And so in this case, I have three state variables of herbivores, coral, and algae, and then the external fishing pressure, which is removing herbivores from the system. And so from this basic model structure, I expanded it to include the herbivore groups that I just mentioned. So now we have our grazers, our generalists, and our browsers. And we also have their two food resources that they consume, the turf algae and the macroalgae. And so again, I'm thinking about whether the dominant herbivore functional group influences the occurrence of alternative stable states, which you assess by looking at these properties of bistability and hysteresis. So these are what I ran analyses for to detect in my model. So I'm going to step through how I did that for each of these. Okay, so for bistability, if we look at like this yellow square up here, so this indicates a run of my model where I set, all of my initial conditions and my parameters, and the two initial conditions that I changed to assess in my bistability analyses were initial coral cover and fishing pressure. So up here, we have a fishing pressure of about 0.5 and an initial coral cover around 0.6. And the yellow color here is then indicating when I ran this model to equilibrium, what was the final coral cover in that system. And so the yellow color here is indicating that in this case, the reef ended up in a high final coral cover state, which is indicative of a healthy reef. And so I also had runs where, in this case, I had a fishing pressure, again, the same of about 0.5, but the reef started with a much lower initial coral cover condition. And then the dark purple here indicates that at equilibrium, that reef ended up in a low coral cover state, indicated by the purple there. And so I did these analyses for many combinations of fishing pressure and initial coral cover conditions. And in order to look for bistability, and what you're looking for to detect bistability here is you're really looking for these ranges here, where for a single level of fishing pressure, in some instances, the reef ends up in a low coral cover state, and in some instances, the reef ends up in a high coral cover state. So this is the range of bistability in this case. And then for my hysteresis analyses, again, hysteresis is thinking about those tipping points, whether or not you are shifting from a healthy to a degraded state, or if you're going from a degraded state and trying to recover back to a healthy state. So that's what I did in my model. I did these forward and reverse runs, where for the red line, this thick red line here, I started with my model and the system had high initial coral cover and low fishing pressure, and I slowly increased the fishing pressure, and then looked at where that tipping point occurred, where the system switched from a high coral cover state to a low coral cover state. And then my reverse runs, I did the opposite. So I started with reefs that were in a very low coral cover condition, had high fishing pressure, and then slowly reduced fishing pressure to then see where the tipping point occurred, where then that system was then able to recover back to a high coral cover state. And so I did these bistability and hysteresis analyses for my model. And again, I was thinking about herbivore communities. So I did these analyses for my model based upon 4 different herbivore community scenarios. The first scenario was basically just kind of a baseline where the herbivore community was split evenly between the 3 groups of herbivores. So my grazers, browsers, and generalists. And then my other 3 scenarios were where one of those groups of herbivores was dominant in the community. So again, 4 scenarios where either the herbivores were evenly split or whether the community was dominated by generalists, browsers, or grazers. Okay, and so here are my results from my bistability analysis. And so again, the 4 different panels are the 4 different herbivore community scenarios that I considered, and we'll step through these one by one. So in this first panel, we can see that when the herbivore communities evenly split between the groups, we see that bistability exists for fishing pressures between about 0.42 to about 0.58. And you can kind of think of this as like a baseline scenario for this modeled coral reef. If instead we change the herbivore community to be dominated by generalists, we see a similar but slightly contracted range of bistability compared to the initial baseline scenario. If we change the herbivore community to be dominated by browsers, we see that in this case, there is the narrowest range of bistability. And when we saw this, this intrigued us, and we wondered if we were able to push it such that we could get bistability to disappear completely. So we increased the dominance of the browsers in our herbivore communities, and we found that if we did increase it enough, we actually saw that bistability disappeared completely, and instead you just had a simple threshold shift in this modeled system. And then for a grazer-dominated community, we see that these communities have the largest range of bistability, and that bistability exists even when there's no fishing pressure happening in the system anymore. So overall, we saw that the herbivore community that is present does influence the fishing pressure conditions when 2 stable states exist. Okay, and so now along the bottom here, I'm showing you the results from a hysteresis analyses, where again, the 4 different panels are the 4 different herbivore community scenarios. And we'll step through these one by one, but they are very similar, basically the same as what we saw for the bistability. So again, in this baseline scenario, we saw that the degradation and recovery trajectories differ from each other. So we see that hysteresis does exist in this system. In a generalist dominated community, we see a similar range of hysteresis, but it's slightly contracted compared to the baseline scenario. In a browser dominated community, again, we saw this narrowest range of hysteresis. And again, if we increase the dominance of the browsers in the herbivore community, we saw that the hysteresis would disappear entirely. And then finally, for a grazer dominated community, there's the largest range of hysteresis, and recovery doesn't occur even when fishing pressure is completely removed from the system. And so as we saw with bistability, we also saw with hysteresis that the herbivore community is influencing the degradation tipping points and the paths to recovery that you can get in these modeled coral reef systems. Okay, so just to summarize, we found that herbivore community composition influences the range of fishing pressure conditions where bistability and hysteresis occur. And so coral reefs with a browser-dominated fish communities are less likely to exhibit alternative stable states in response to fishing pressure, whereas coral reefs with grazer-dominated fish communities may not recover to high coral cover states even if fishing pressure is entirely removed from the system. And overall, found that incorporating functional groups into a coral reef resilience model can provide insights about conditions causing alternative stable states, which can be important for effective management of these systems. Okay, so I told you about two projects thinking about herbivorous fishes on coral reefs. And so now I'm going to shift gears and talk to you about nutrient addition effects in coral reef systems. And so I've been involved in a variety of projects looking at nutrient addition effects on algal growth, interactions, and physical characteristics. I'm only going to tell you details of one of those projects, and then I'll just briefly mention the other ones. And so the project I'm going to tell you about is one where we were thinking about how macroalgae and nutrients may influence the growth of turf algae on coral reefs. And so, as I mentioned earlier, they have the broad classifications of algae as turf algae versus macroalgae. And in terms of turf algae on reefs, another point is that the composition of turf algae is still unclear in reef systems, although there is ongoing research to try and figure that out and clarify it. But it has been recognized that the length of turf algae that you find on reefs can be important. And so specifically, for thinking about turf algae, you can think about spats versus LSATs. And so spats are short productive algal turfs that are less than 5 millimeters in height, and LSATs are long sediment-laden algal turfs that are greater than 5 millimeters in height, and as the name suggests, typically have sediment associated with them. And so it's long been considered that when reefs shift to a degraded state, that typically means that you're going to find a lot of macroalgae on those reefs. However, more recently, there's also been the acknowledgement and recognition that a degraded coral reef, it could have lots of macroalgae, but it's possible that it could not have that much macroalgae and instead could have a bunch of these LSATs, or these long sediment-laden algal turfs. And so this has spurred a lot of research into understanding what is contributing to the development of these LSATs on reef systems. And so two common impacts on reefs are reduced herbivory and nutrient enrichment. And so we know that our previous research had shown that reduced herbivory can contribute to transitions from spats to LSATs. And also you can have reduced herbivory caused on reefs by increased sediments. And so when you have LSATs, so turf with lots of sediments, that may actually compound reduced herbivory by preventing herbivores from feeding on that turf algae. And then it is also known that nutrient enrichment often increases turf height, although there can be some interesting interactions with herbivores when you add nutrients to algae. And so for this study, one thing we noted is that the same factors that are known to promote the growth of turf algae, so reduced herbivory and nutrient enrichment, also often promote the growth of macroalgae. So if you have a reef that's experiencing reduced herbivory and nutrient enrichment, and you're seeing more long turfs and you're seeing more macroalgae, this really led us to the question of, well, how is the macroalgae then affecting the turf algae that you find on the reef? And so when we looked in the literature, we found that algal-algal interaction studies were actually relatively rare on coral reefs. There had been a few looking at interactions between different types of macroalgae, but at the time, there hadn't been any previous studies that looked at the effects of macroalgae on turf algae in tropical coral reef systems. And then in the lab that I was in, we also always think about the effect of nutrient addition. So we also wanted to ask how nutrient addition may affect the interaction between macroalgae and turf. So that was our question then, was how do nutrient enrichment and macroalgal presence affect turf height and sediment accumulation? So we set up a two-factor experiment to assess this, where we went out and found coral bommies that were covered in turf algae, and we set up our plots and we caged them, and then we applied our treatments where we manipulated the absence or presence of macroalgae and the absence or presence of additional nutrients in this system. And then we assessed the turf height and the sediment depth in that turf algae over time. And here is a figure of our results showing you our turf height over time. And so we specifically calculated differences from initial for turf height. So that's why on day one, it's all 0, because we looked at basically the difference in turf height related to what it was on day one. And so the main takeaways here are that turf height is positively and independently affected by macroalgal presence and nutrient enrichment. So specifically, turf height increased 127% more with nutrient enrichment, and it increased 34% more when macroalgae were present. Okay, and then these are our sediment depth results. And so here the main takeaway is that sediment depth is negatively affected by macroalgal presence. And specifically, sediment depth accumulation decreased by 58% when macroalgae were present. And so here's just a summary of the results. So again, we found that turf height was increased by the presence of macroalgae and by the addition of nutrients. And we saw that sediment depth was decreased by the presence of macroalgae, and there was no effective nutrients, and there was no interaction between presence of macroalgae and nutrient addition for either of our response variables. So from the study, we actually found a novel facilitation where macroalgae can facilitate the growth of turf. Since macroalgae and turf algae are both algae and they compete for similar resources, we kind of went into this suspecting that macroalgae may actually have a negative effect on the turf algae around it. So we were kind of surprised to see this facilitative effect. But the implications of this are that having macroalgae may actually help promote shifts from short, healthy turf to longer, less desirable turf states on coral reefs. So could help with this transition from spats to LSATs. And one point I do need to make is that for this experiment, we caged all of our plots, which excluded herbivores from them. But on coral reefs where herbivores are going to be present there, if you want to, think about what the effects would be if herbivores have access to that macroalgae, whether or not this facilitative effect exists for macroalgae is likely going to depend upon the type of macroalgae that's present on the reef and its palatability to the herbivores that are also present on the reef. Because if it's very palatable to the herbivores, herbivores may just come through and consume it, and then you're not going to see this novel facilitation happening. But overall, we did find that macroalgae and nutrient addition may each lead to positive feedbacks that could stabilize algal-dominated states on coral reefs. Okay, and so again, I've been involved in some other studies thinking about nutrient addition effects on coral reefs. And so we've found some interesting things where we found that with added nutrients, the physical characteristics of macroalgae can change, sorry. So we actually saw that like the calcification can increase or the toughness can increase of macroalgae. And so these are physical characteristics that may then influence the palatability of those macroalgae to herbivores. And then also involved in a study where we were thinking about the timing of nutrient availability on reefs. So thinking about like press versus pulse nutrient regimes and how that may affect macroalgal growth and also macroalgal interactions on coral reefs. And if anyone is going to Benthic Ecology meeting next week, this is the project that I'm going to be presenting on there. So if you want to hear more about that, then you can come watch my talk. Okay, so coral reefs are shifting to degraded algal-dominated states, and we know that anthropogenic impacts are contributing to these shifts by modifying top-down and bottom-up processes within reef systems. In terms of top-down processes, we know that herbivore functional redundancy and complementarity support reef resilience, and we found that it can be important to assess the functional roles of herbivorous fishes within specific reef systems. And we also know that fishing can modify the top-down process of herbivory by altering herbivorous fish communities. And we found that changes in the herbivorous fish communities may actually affect reef resilience by modifying those conditions when alternative stable states occur. And then in terms of bottom-up processes, coral reefs typically exist in nutrient-poor waters, but humans are modifying the availability of nutrients with extra inputs into reef systems. And so we found a variety of different things in terms of the impacts of nutrient enrichment such as that nutrient enrichment may promote shifts from short turfs to long turfs. It may also promote macroalgal growth, which could then facilitate turf growth. We found that nutrient enrichment can alter macroalgal physical characteristics, which could make them less susceptible to herbivory. And then finally, we have some data to suggest that nutrient enrichment may influence the benthic macroalgal community composition on coral reefs by changing the interactions between different macroalgae. Okay, so that was my PhD research in a seminar format for you. So now I'm just going to pivot for the last couple of minutes and talk about some of the projects I'm involved in here at Dauphin Island as a postdoc in the Center of Excellence. So one of the big projects that we have going on now is our habitat connectivity project, where we are assessing whether habitat connectivity between seagrasses and salt marshes influences secondary production of nearshore coastal habitats. And so we are interested in both seagrasses and salt marshes here. So we have two kind of sub-questions related to this overarching objective. The first one being that is secondary production of salt marsh habitats affected by the presence of adjacent seagrass habitats? And then we have the seagrass equivalent of that question. So is seagrass production of seagrass habitats affected by the presence of adjacent salt marsh habitats? And for this project, we have sites that span both Mobile and Baldwin County. And this was our study design in terms of when we went out to go select sites for this study. So we looked for seagrasses that were isolated from salt marsh habitats. So we looked for seagrass beds that either were near a shoreline that was sandy, or we found seagrass beds that were kind of in the middle of a waterway, so they weren't super close to a shoreline that had salt marsh habitat on it. On the other side, we also found marshes that were isolated from seagrass habitat, so then just found marsh habitat that did not have a seagrass immediately adjacent to it offshore. And then we have our adjacent treatments where we found marshes and seagrass beds that were adjacent to each other, and we set up both a marsh adjacent site and a seagrass adjacent site. And in this case, all of our sites, we just define them as a 30-meter by 2-meter area for these different site types. And so here's just a map showing you where our sites are over in Baldwin County. And so in Baldwin County, we are only addressing the seagrass question for this project. So we have seagrass sites where the seagrass beds are adjacent to a salt marsh habitat and seagrass beds that are isolated from a salt marsh habitat. And here's a map showing you our sites in Mobile County. And so in this case, we are addressing both the seagrass and the salt marsh questions. So we have all 4 site types kind of spread out around here. And here's just some photos showing you some of our sites. You can get an idea of what our marsh sites look like. And, you know, in our marsh and seagrass adjacent sites, you can kind of see what they look like. I realize the photos of the seagrass sites are just pictures of water. You'll have to trust me that there is seagrass there. We did go out and check. So there is seagrass under the water at these sites. And so to go out and sample these sites, we are, oh, the video's got a little smushed. But we are going out to these sites and doing suction sampling at them every other month for this year. And so we're really trying to get at what are the small invertebrates that are present in these habitats and in these sites. And is that influenced by whether or not they are adjacent to the other site type? And so we'll be looking, you know, at like the diversity of the invertebrates that we collect and also calculating their secondary production. Sorry, the video's got a little messed up, but there's a lovely YouTube video of Ken Heck explaining suction sampling. If you haven't seen it and want to get a full idea of the picture, I just grabbed some snippets from it for this. Okay, so the Habitat Connectivity Project is one of the big ones we have going on, but we also have some work going on where we're trying to assess the effectiveness of doing eDNA sampling for detecting fishes around here. And so we're comparing eDNA samples to the trawls that are done. And we also have a collaboration with the Smithsonian Marine Geo Network, which involves a number of different projects, but We also committed to doing annual sampling of seagrass and salt marsh habitats, again, in both Mobile and Baldwin County for the next few years. So if anyone is interested in learning more about these projects or wants to talk to me about them, please come talk to me. And with that, I have many collaborators to thank and funding sources. And I would like to thank all of you here and all of you on Zoom for your attention today. And with that, I will take any questions.
Dana. So it looks like we have about 10 minutes left for questions. So ask away, and I think Brandy's on Zoom, so she can get if there are any Zoom questions.
I feel like this means either I explained everything very well or terribly.
Just like a curiosity question, yeah. The videos was there a person underwater videoing for a certain amount of time, or like cameras set in water for
yeah. Yeah, so the question from the audience here was for the underwater videos, whether or not there was a person present or whether a camera was just kind of left out for a while. And so it was the latter. I would put a camera out. I think I left them for I think it ranged between 4 to 6 hours, kind of depending on the day. But yeah, there was not a person there. So trying to minimize, having a human observer there and affecting the behavior of the fishes.
Great Carmichael online asks, it's a great presentation. How do you think shifting baselines can affect your Hysteresis model and expectations?
Yeah, good point. I mean, shifting baselines definitely will affect many things. Yeah, I think to get at that with the model, it would really need to think about maybe trying to model specific reef systems. So in this case, it was just kind of a generalized coral reef model. I didn't parameterize it specifically for a specific reef like in Moorea or anywhere around the world. But if you were going to apply my model to a specific reef system and try to get at looking at hysteresis and bistability, I do think it would be important to consider what conditions you are parameterizing it for. So if you're looking at present day and whether or not present day is a good indication of maybe what you would consider a healthy coral reef. And so, yeah, so really thinking about the different states that you are interested in and how those may differ present day versus maybe historically would be important to consider in that case. So definitely going to have an impact in terms of if you're thinking about specific reef systems.
Yeah. So on your the macroalgae, your first experiment, you had a variety of algae. Some were brown. I saw halibut down there, which is the green. Physiota, which is known to have chemical defenses. Do you think if you chose different algal communities, you might have seen different results?
Yeah, and that is something that yes, definitely. So if in the paper, that was definitely something that we heard from the reviewers is you really need to consider the context in terms of drawing conclusions for the system. So if you do read the paper and, I was trying to say it in terms of the conclusions that we are making, trying to do it in the context of what we did in this system at this time. Because yeah, if you offer different choices than what we did, they may have exhibited different preferences. So in terms of thinking about that, in terms of, I talked about how I think we saw that in some cases, the fish species we observed performed a different role. So exhibited a preference for a different macroalgae than we saw in previous studies. I have a whole table in the article where we were thinking about that. And we did really try to look and see whether or not there were similar choices offered. And so obviously we're not going to have the exact same macroalgal choice assay in our experiment as was done in the other places, because it's going to depend upon the macroalgae are there. But we at least did try to see if the species that they preferred in our study was also offered in other studies before like trying to make any conclusions like that. And
I was shocked that the Calamita was not really fed upon, I guess.
yeah, I mean, and so, you know, another thing is that study was done on a fringing reef in Moorea, and the fringing reefs there are fairly impacted already. So the herbivore community is also going to be less diverse and smaller than what you would see on other reefs in Moorea. So, you know, go back and do my PhD for another 5 more years or whatever. I would go out and, you know, do some choice assays like that on like the back reefs and on the fore reefs, and also just get an idea how in one reef system, how those preferences could change and the communities could change from like a fringing reef to back reef to fore reef. But yeah, definitely very context dependent. So yeah. Yeah.
For your reading addition feeding study, did you have any controls or any page controls? Like, so did you only have experimental plots?
Yes, and
Because you also were excluding grazers, right? So
yep.
Okay, so can you draw the conclusions you're drawing without knowing if those conclusions aren't just because you're excluding grazers or because of the page itself? How did you address that?
So we are, so for those online, the question is about if we assessed like caging effects, or if we had, you know, any control plots that didn't have any sort of treatments or cages applied to them in the third study that I talked about. And so we did not specifically have control plots for that study at that time, but my advisor has done a lot of studies like that, where they have done those assessments in terms of looking at the effects of having cages out there and if that affects turf growth. And there has not been evidence of that. And so I'm pretty sure we did this in the paper, but if we didn't, we've certainly addressed it in other studies as well. There is other research done in Moorea in those systems that assess those things. And so, you know, one of the main things that we specifically would try to do for those studies is like, for measuring the turf algae in the plots, we wouldn't measure turf algae right near the edge of a plot, you know, because we like right next to where the cage was pounded in. You would try to measure it kind of where the turf algae wasn't potentially going to be physically up against caging material. So that was, you know, one thing that we did, but also my advisor has done previous research showing that the cages that we use were not negatively impacting that community. And then in terms of the herbivores, yes, so we caged all of our plots to specifically exclude the effect of herbivores. So that is a big, you know, caveat for that study in terms of thinking about it. And again, in the paper, we have like a discussion paragraph kind of doing a thought experiment of like, well, if you have herbivores that are present, is this likely to occur? And that's where kind of thinking about the macroalgae that are present and its palatability to the herbivore community that is there is likely going to influence whether or not you could see that facilitative effect.